أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم انطلقوا إلى ما كنتم به تكذبون انطلقوا إلى ظل ذي ثلاث شعب ولا يغني من اللهب إنها ترمي بشرر كالقصر كأنه جمالة صفر ويل يومئذ للمكذبين صدق الله العلي العظيم. Lecture number 11 for 11th class, first year class. Cytoskeleton. The skeleton is provided to each part of the living thing. Skeleton was provided to us in the form of the bones. Skeletal system consists of bone and cartilage. You will study in second or third chapter of second year in much detail cartilage and the bones, how they are composed and how they are supporting you. But there are some things which are required to support the cell. Similarly, unit part of the body, nature did not leave alone the unit part of the body cell. The single unit means cell was also provided with cytoskeleton. In the last lecture, I was discussing one part of the cytoskeleton, microtubules, microfilaments. Now I come back to second part, which is a microtubules. You know, cilia were composed of the microtubule we were discussing in the last lecture. The microtubule is a polymer. The large molecule is called polymer. Small molecule is called the monomer. Small molecule combine to form the large molecule. Water molecule is released. This is called condensational polymerization. So what is condensational polymerization? The small molecules combine, monomers combine to form the large molecule, and large molecule is the polymer. So microtubule is example of polymer, one type of polymer. Starch is the polymer. The protein is the polymer, amino acid plus amino acid. Protein plus water, condensational polymerization. So tubulin is the monomer of the microtubule, means small molecule which combine to form large molecule. Water is released in this example microtubule. Which thing we are studying? Cytoskeleton. Which part of cytoskeleton? Second part of the cytoskeleton. Name this type of cytoskeleton microtubules. How many type of microtubulins? Tubules mean how many type of tubulins are present? Two type of tubulins are present in the microtubule. Is there any difference between the composition of the cilia and this one? No, that is same microtubule. Is this any difference between spindle fibers which are present in mitosis, meiosis? They are composed of microtubules also. Is this microtubule is of same type? One type of microtubule, alpha beta tubulin, same type of microtubules which are present in spindle fiber, which is present in cilia, which is present in flagella. So that is the part of the cytoskeleton that is giving strength to the cell, doing other functions as well. So two type of the tubulin present in microtubule. Three type of cytoskeleton I will speak once again. One is microfilament, we were studying in the last lecture. Repeat the lecture number 10 and see what is the microfilament. In this lecture I am telling you the type of cytoskeleton which I left. One is microtubule and third one is intermediate filament, which I have to discuss after some time. Microtubule is a polymer. It is a type of cytoskeleton. It is composed of two type of tubulin, alpha tubulin and the beta tubulin. Alpha and beta tubulin. One is alpha tubulin and one is the beta tubulin. So alpha and beta, two type of tubulin. After some time I will show you diagram in which I will show you that alpha and beta tubulin, they are combining to form the microtubule. We go to the slide. Intermediate filament, the third part of the cytoskeleton. What is the cytoskeleton? Once again, it is a type of the protein. Why we are discussing now? Because that is the protein which is present in the form of several macromolecule or the polymer in the cell to give strength to the cell with some additional functions. Why we are naming vimentin, why we are naming tubulin, why we are naming microtubules, why we are naming microfilament? Because this is cytoskeleton composed of the protein, but these names are quite different. These all are types of the protein. For example, vimentin you are watching in the slide. The vimentin is another type of the protein, but it is present in the intermediate filament. It is present in intermediate filament. Intermediate, third type of the filament. Why it is called intermediate filament? The size of the protein, filament, why, what is filament? When protein molecules, protein fibers combine, they form a fiber. When protein molecules combine, they form a filament or the fiber. That filament is the, definitely that is the filament, intermediate filament, microfilament, microtubule. So protein molecules combine to form a fiber type structure like cilia or flagella, that is called as a filament. So why this third type of cytoskeleton is called intermediate filament? The reason is that because size of intermediate filament is just like between, not like, it is between microtubule and microfilament. Still we are confused. Still we are confused. What is the function of microfilament? So we forgot in the last lecture. Microfilament, actin and myosin, that is present in the muscle cell. The muscles contract due to the microfilament. You know, three type of proteins were present. One is actin, one was tropomyosin, third was troponin, and that was a complicated mechanism. In the senior classes I will teach you. When actin and myosin attach with each other, just like two fingers attach, when they attach, the muscles contract, muscles relax, then muscles relax. This contraction, relaxation move the body. We become motile. Our organs become motile. The muscles are involved in with the motion, They move over forelimb. They move over hand limb. Our body structures are being moved by skeletal system because skeletal systems are attached with the muscles. Cardiac muscles are pumping the blood from the heart. Similarly, the muscles in the esophagus pushing the food into the stomach. Similarly, the muscles in the arteries are pushing the blood to the different parts of the body. So muscles in the pharynx pushing the food and even the air towards the lower part of the body. So these muscles can work only when the protein inside the muscles will contract, and protein will contract only when actin protein, which is microfilament. I am just giving you meaning of first type of skeleton, microfilament cytoskeleton. If you have forgotten, microfilament, actin protein. Myosin I did not explain, but I am telling you role of actin. One role of actin is that it will combine with the myosin, muscles contract. Other role I told you in the last lecture, that the actin filament will move the cytoplasm. It will move the cytoplasm. When it will move the cytoplasm, what will be the benefit of moving the cytoplasm? That cytoplasm will move, the liquid part of the cytoplasm will move, cytosol will move, and molecules in the cytoplasm will be able to transport from the one part of the cell to another part of the cell. So motility of the cell, cytoplasm will move. Now I am telling you the third part of cytoskeleton. You are asking second type of just explain. Microtubule composed of tubulin, second type of cytoskeleton. Example of microtubule, yes, I can give you cilia, flagella. We just studied in the last lecture. Cilia, I was talking about the 9-triplet microtubule, which is a polymer. And function of another function of microtubule, yes, I can tell you. Spindle fibers are composed of the microtubule. Spindle fibers are not formed, the cell will not divide. No question, sir. They are not completely, cytoskeleton is not giving strength to the cell. It is also doing additional function. Yes, I told you. Additional function, I am telling, additional functions like cell division, contraction of the muscles. These are also the movement of the cytoplasm. These all functions are strengthening the cell. They are making the cell strong. In that sense, we can say also cytoskeleton. So third type of the cytoskeleton, Vimentin, V-I-M-E-N-T-I-N, spelling is written. It is the intermediate filament. The size is intermediate, mean that is middle size between the microfilament and the microtubule. A true skeleton, if you are questioning, sir, which type of the cytoskeleton undergoes exact right definition of cytoskeleton, because meaning of cytoskeleton is to provide the strength to the cell. To which fiber, which skeleton is providing the strength to the cell? I will tell you, yes, this one is that. This one, the intermediate filament with Vimentin protein is giving mechanical strength, the true cytoskeleton, exact cytoskeleton. Other cytoskeleton, microtubules, microfilament are doing some additional functions. So we'll go back to another slide which I want to show you. This one is a dynein arms. Dynein arms, main protein between outer and inner fibril who move cilia and flagella. Before reaching this slide, I would like to show you one diagram. Then I will tell you this one. because you know that example I have to tell you. Just hold on. And dynein arms I may not discuss in this lecture. Reason is that dynein arms is present in the structure of cilia. I want to show you the diagrams now, and the diagrams of the cytoskeleton. Three type of the cytoskeleton. Look at these three diagrams. These are very important. I have to highlight those. Three type of cytoskeleton is visible to you. Yes, this is the cytoskeleton. Three type of cytoskeleton visible to you. This is number one, the first type of cytoskeleton. This is the second type of cytoskeleton. So two cytoskeletons are visible to you. I will explain these two first of all. If you remember the last lecture, I told you that a microfilament consists of three protein. Central protein, actin, visible? Yes, this is actin. Central protein. Second protein which is present, which is surrounding the actin, consists of two chains of actin which are twisted upon each other. Similarly, tropomyosin with little gray color, you can see. Two chains are present in tropomyosin. They are present above actin, and they are also in a doublet form, mean they are also twisted, a double chain, and they are twisted upon each other. The third chain, which is the troponin. Three chains are superficial chains because these are the chains which will, due to which actin filament in the muscle cell composition, two filament, actin filament, myosin filament. Actin filament combined with myosin filament, muscles will contract. The muscles will contract. So what will happen? This actin filament and myosin filament, they will contract. And third one is troponin. If you see troponin, that is very important. Look here, one part of troponin, second part of troponin. I will rub this one because I just want to show you troponin once again. Yes, I close this one. I would like to show you troponin. Student, this is one troponin, this is another troponin. There is the gap between troponin. What is the objective to tell you the significant point? There is no continuous layer in case of the troponin. So what are two continuous protein? Actin is a continuous protein. Troponin, myosin is a continuous protein. But troponin is not a continuous protein, This objective, why the troponin is discontinuous in the muscle structure, in muscles contraction and relaxation, you will understand the logic reasoning of this, why troponin is not continuous layer. But this is amazing structure that one protein is in the center, just in a twisted format, and then tropomyosin. If I will tell you simple sentence, you will think how this is formed. Look here. Troponin is just a door. Troponin can move. I will teach you in muscle cell, but one point I just wanted to share. And don't worry if you will get a little complications there, because you will study much in detail in the muscle cell. Troponin, if you will remove little of it, this troponin you will remove. Who will remove? Calcium ions will remove. That's why I was talking about calcium ions were also very necessary. Parathormone is very necessary. Calcitonin is very necessary. Calcium ion, calcium gates are present in muscle cell. They will just remove the troponin, and tropomyosin will also move. Why it will move? Why it is move? Troponin attach with the tropomyosin. Troponin move, tropomyosin move, actin exposed. Amazing. This thing I have to repeat now. Troponin is just like hinge-like structure. Yani hinge just like door. Tropomyosin is a door, and troponin is the handle of the door. If you will move troponin, because that is not, a handle is not present throughout the door, that is present partly, and when you will remove the handle of the troponin, the tropomyosin will expose. The tropomyosin will also move. Tropomyosin moves, actin will be exposed, and this actin which will expose, this is important because actin will expose, it will combine with the myosin. So actin is exposed, it will combine with the myosin, and then the muscles will contract. It means troponin, tropomyosin are so important that if they are not properly working, calcium ions even not coming, that is not moving the troponin, and troponin is not moving the tropomyosin, actin is not exposing, the actin will not combine with the myosin. When actin will not combine with the myosin, muscles will not contract. And muscles will not contraction and relaxation will not take place, so it will affect all the body of our body. Go back to microtubules. It was simple. Microtubule is comparatively simple. in the sense because it is, I should magnify this one for you. I think this is a extensive maximum magnification. Look at, this is the microtubules cytoskeleton. It consists of two type of tubulin, alpha tubulin. Alpha tubulin, one is a alpha tubulin and one is a beta tubulin. You are watching two colors, one is a alpha tubulin, one is a beta tubulin. Intermediate filaments, this is the intermediate filament. Vimentin protein twisted format, in the twisted format. An intermediate filament function is very easy. How you can support intermediate filament function? They are just giving support to the cell. I am once again telling you the function of all filaments. Easiest function, support. Protein formation, microtubule, cilia formation, flagella formation. Cilia structure I am going to teach you. I know, you know, I use the word dynein arms. Why I stopped to teach you dynein arms? Because that is present in structure of cilia. That is a logical conclusion how the cilia move. Because you normally see the cilia small fiber, even the nose small cilia fiber, even trachean small cilia fiber, small locomotive structure in bacteria. They are not simple. If you will see cilia, this is just a full world. How they are moving. There are a lot of proteins working just like a full factory. Machines are working to move single cilia. That is my next lecture, in which I will tell you how the cilia and flagella move, because both structures are almost same. Dynein arm is one type of protein that is very important protein, because that is helping in movement of the cilia. We go back to the microtubule, involved in the formation of the spindle fiber, involved in the formation of the cilia, involved in the formation of the flagella. We'll go back to next one. Now this is the one, last one. The microfilament. The slide is visible, the microfilament, and this is quite complicated at your level because you will study this one in the second year. But once again I will tell you, one is a central actin, then tropomyosin full, actin full, tropomyosin full. Both are double strand, both are twisted, both are double chain, and then the partly troponin handle of the door. Troponin move. Who will move? Calcium ions in the muscle. In the muscle cell, I will teach you that calcium gates will open, calcium ions will come, it will move the troponin, tropomyosin move, and actin filament exposed, combined with the myosin, muscles contract, you move. Gentlemen, your movement is also very technical. Thank you very much. This is lecture number 11, and wait for structure of cilia. Allah Hafiz.